Good morning, everyone, and welcome to this online platform. Today, we'll discuss about physical pharmaceutics second practical experiment number 11. So, aim of our practical, that is to determine accelerated stability studies. Stability study means these are the process used to evaluate how product, pharmaceutical or food product changes over time under various environmental conditions. It helps to determine shelf life and appropriate storage condition for a product by assessing its physical, chemical, and biological properties. Accelerated stability study means increase the temperature, such a temperature that temperature product, pharmaceutical product or food product have stable or not stable that can be found by physical, chemical, and biological properties evaluating. Once they are stable in different temperature conditions, that product is stable, but that is suitable, means that directly affect to the safety of drug. So this accelerated stability study means to determine pharmaceutical product by increasing or elevating trace conditions, mean increasing the temperature and assessing the physical, chemical, and microbiological stability over a defined period. So accelerated stability studies, these are the study will carry out different temperature conditions and evaluate physical, chemical, microbiological that product is stable or not. So objective, that is to evaluate stability of pharmaceutical product under accelerated environmental conditions, means increase, accelerate means increase temperature conditions for predict its shelf life and determine quality, safety and efficacy over time. The material requires stability chambers, drug product, final packaging, tablet, capsule, syrup. Stability chambers is required for study this purpose with controlled temperature and humidity. Analytical instrument also required, HPLC, for assessing of impurities, UV visible spectroscopy. For physical properties determines pH meter, determine the pH, dissolution tester, car pressure titration for moisture content, microbial testing apparatus. So these are the stability chamber. Stability chamber can control environment chamber used to stimulate various environmental conditions, primarily temperature and humidity, to assess the stability shelf life of product, especially in pharmaceutical industry. So stability chamber, it can control environmental temperature is rising, real world conditions, different temperature you can rise in this chamber and you can assessing. So in that components of stability chamber, in that control panel, electronic electrical components, door, cold air inlet vent, tray, hot air outlet vents. So this is the stability chamber. This stability chamber, in that we increasing temperature, different temperature conditions have maintained. Primary temperature and humidity, assess the stability conditions. ICH guidelines. So this stability study will carry out ICH guidelines as well as World Health Organization guidelines. ICH stand for International Conference on Harmonization. These guidelines, technical requirement for a registration of pharmaceuticals for ICH is a unique project that develop technical requirement for new drug product for facilitate the registration drug regulating authorities. This ICH secretariat is situated Geneva, Switzerland. The ICH is a joint initiative involving both regulatory and industry as equal partners in the scientific and technical discussion, which are required to assure and assess the quality, safety, and efficacy of medicine. These are different zones, climatic zone association with conditions, storage conditions, accelerated, intermediate, long term. Parameter to be tested: tablet, capsule. Tablet evaluation test, that is appearance, weight variations, hardness, disintegration test. Moisture content and liquid syrup: color, clarity, pH, viscosity, microbial limit, degradation product. ICH guidelines: Q quality topics, S safety topic, E efficacy topics, M multidisciplinary topic. Quality: these are related to chemical pharmaceutical quality assurance, stability testing, impurity testing. Safety: efficacy and multidisciplinary. So applications in that different guidelines called ICH guidelines, that is Q1. Q1A, that is stability testing. Q2 analytical method validation. Q3 impurity testing. Q4 pharmacopoeia. Q5 quality of biotechnological. Q6 specification of new drug substance. Q7 good manufacturing practice. Q8 pharmaceutical development. Q9 quality risk management. Q10 pharmaceutical quality system. Q11 development and manufacture of drug substance. Q12 life cycle management. Q13 continuous manufacturing of drug substance. And Q14 analytical process development. So this ICH guidelines is combined different countries to form unique, which is suitable for all over the countries, the stability guidelines. So these are the different guidelines: Q1 that is stability testing, Q2 that is analytical method validation, and Q3 that is impurity testing. So applications of ICH guidelines, that is drug development. ICH efficacy guidelines: E series guide, design, conduct, safety, and reporting of clinical trials. Good clinical practice: improve the consistency and reliability of clinical data across countries. Quality control: Q series cover manufacturing process, specification, and pharmaceutical quality system. Ensure consistent drug quality and compliance with international standard. Safety assessment: regulatory submissions, risk management and pharmacovigilance. Global harmonization: promote international cooperation among the regulatory agencies and pharmaceutical industry, reducing duplications and accelerating global access to new treatment. Advantages: global harmonization, improve drug quality, safety, and efficacy. Cost reduction, regulatory predictability, enhance collaboration. WHO guidelines for pharmaceutical product. World Health Organization provide comprehensive set of guidelines for pharmaceutical product, ensuring the quality, safety, and efficacy throughout the product life cycle. These guidelines developed through the global consultative process. Good manufacturing practice, quality assurance, regulatory standard, specific guidelines, sterile pharmaceutical production, research development, facility and quality control, laboratories. So any dosage form, vaccines, pharmaceutical dosage form, food product stability is required. To stability is a safety of drug, to assessing the safety of drug. So accelerated stability studies stimulate long-term storage conditions, shorter period, help and determine the product expiry date, ensuring consistent product quality. Means simply, it can identify the expiry date of product or shelf life. Hope you can understanding this practical. Thank you. Have a good day.